Hi, I'm Paramita Chakrabarty. I'm joined by two of my trainees, Dr. Zachary Sorrentino and Ms. Tasha Williams, as we discuss our current publication in brain communications. The premise of our study is to explore the heterogeneity of neuropathology that you often find in Alzheimer's disease and related dementias. For example, if you look at different population cohorts such as the ROS-MAP, the MRCC-FAS, and others, you'll see that in addition to the Alzheimer's typical A-beta and tau pathologies, you get robust co-occurring alpha-synuclein pathology in as many as 30 to 40% of these patients. It is possible that these mixed pathologies may not only lead to molecular diversity of the biologically active seeds in the brain, but they might also lead to clinical heterogeneity as well. In this study, we specifically investigated whether the cofibrillized polymers of tau and alpha-synuclein possess unique cross-sitting properties compared to the individual fibrils themselves when tested independently in a mouse model of tauopathy and a mouse model of synucleinopathy.
My name is Zachary Sorrentino. The first part of this research study, really what we did was replicate an in vitro study from Dr. Giason and Dr. Virginia Lee back in 2003, when they showed that alpha-synuclein can induce tau to aggregate in vitro without the help of any cofactors. We reproduced these initial tau alpha-synuclein copolymerization experiments in our lab, which is shown in this electron micrograph picture, where gold nanoparticle beads of different sizes are bound to different antibodies, one specific for tau, the other specific for alpha-synuclein, showing that these mixed copolymer fibrils are decorated by both sizes, indicating that. And we tested the unique cross-seeding properties of these mixed polymers compared to individual tau or alpha-synuclein fibrils in HEK cell culture, which themselves were expressing either full-length tau or alpha-synuclein. And in this seeding assay performed in these cells, we found that when full-length tau is expressed, then the copolymers of tau and alpha-synuclein together dramatically enhanced tau seeding compared to cells treated with tau fibrils or alpha-synuclein fibrils alone. On the other hand, tau and alpha-synuclein copolymers together did not boost alpha-synuclein seeding relative to alpha-synuclein fibrils in an identical assay with alpha-synuclein. So this indicates that these mixed fibrils have unique prion-like properties depending on the protein that they are themselves seeding.
Thank you, Zach. I'm Tasha Williams, a graduate student in the lab of Dr. Chakrabarty. And here we then tested the efficiency of these copolymers and individual fibrils in the PS19 mice, which are widely used as a model for tauopathy. Using a neuropathological scoring system shown in the scale here, we found that intrahippocampal injection of copolymers as well as individual fibrils in these mice robustly induced phosphorylated tau in both the ipsilateral and contralateral hemispheres. The graphs below show the burden of phosphorylated tau for the different fibril-injected mice in the ipsilateral and the contralateral hippocampus, as well as the cortex. Using the same scoring criteria, we quantified the extent of transmission of human tau cross-seeded by the tau and alpha-synuclein copolymer seeds or the K18 tau seeds alone. We found that the copolymer-seeded tauopathy was more efficient and had a selective propagation along these neuroanatomic connections compared to the K18 tau fibrils themselves. Next, we examined the effect of these fibrils in the M20 mice that expresses full-length human alpha-synuclein. Hippocampal injections of the alpha-synuclein fibrils resulted in robust intracellular alpha-synuclein pathology as expected, and surprisingly, the tau fibrils also induced synuclein pathology, showing cooperativity. However, we found that the copolymers that were very inefficient in seeding synuclein pathology in these mice. The graphs below show the burden of phosphorylated alpha-synuclein for the different fibril-injected mice in the ipsilateral and contralateral hippocampus and cortex. When we compared the transmission efficiency, we found that consistent with inefficient local induction patterns, the copolymers showed lower rates of transmission compared to the alpha-synuclein fibrils themselves.
Our study has a few caveats, such as using recombinant fibrils as opposed to mixed fibrils from patient lysate. And also we only examined a single time point, so we did not see the evolution of pathology.
In summary, we showed that tau and alpha-synuclein copolymer fibrils potentiate the transmission of tauopathy while restricting the spread of alpha-synuclein pathology in the respective mouse models. We believe that our study highlights the cooperativity and synergism between different proteinaceous seeds that lead to unique cross-seeding patterns, and it's significant because of its potential relationship to the heterogeneity observed in Alzheimer's disease and its subtypes. Last but not the least, we would like to thank the Alzheimer's Association, an NIA fellowship for Dr. Sorrentino, and an NIA training grant that currently supports Ms. Williams. Thank you.